to run up on your boy. 엉덩이 빵빵빵 남자들은 탐탐탐 내 입술 냠냠냠 어디 가도 탕탕탕 등장은 짠짠짠 뛰자 뛰자 팡팡팡 디스코 팡팡팡 Hello.
Ladies and gentlemen, may I have your attention please?
Assalamualaikum warahmatullahi wabarakatuh, and hello everyone. Today we are going to proceed to the next chapter in our syllabus. My friend and I are going to explain and present to you about chapter 7, which we are going to look specifically in the biological or biology laboratory. So without wasting time, let's get started. In this chapter, we are going to learn about how to handle the epithelial tissue study, and then we also will look on how we are going to conduct experiment with bacteria and fungi, then special concern in the study of fungi and molds, extraction of chlorophyll and the use of flammable solvent, and lastly, handling of laboratory animals. As biology and physiology teachers, it is vital for us to be familiar with all the following safety guidelines as well as any section of the handbook that are relevant to the teaching program. In other words, if we are going to be a biology teacher or required to handle a biology experiment, so we need to follow all the safety precaution and follow all the handbook that is already handed to us. This is because teaching a laboratory class poses additional risk as compared to conventional instruction in a classroom. So, before we, as a teacher, enter the lab, we must be aware of the safety measures and always be prepared of what experiment that we are going to conduct on that day. Without further ado, let's start with our first objective of this chapter, that is to learn on how to handle the epithelial tissue study. As we learn biology and in the future we are going to conduct an experiment in order to teach our students, human cheek cell experiment is not something that is uncommon among us. It is familiar experiment as it is the simplest way to learn about human cell structure. As we know, in the cheek cell experiment, there will be a procedure that requires a student to scrape the inside of the cheek to get the cheek cell. If we are handling cheek cell study, in order to look the structure of the cheek cell, as a teacher or instructor of the lab, we need to make sure that our student take a great care or carry out a very careful safety measure in order to avoid any accident or injury during pricking the inside of the cheek. We also need to make sure to use just a cotton tip swab or the blunt edge of the toothpick. Usually, we will use toothpick to conduct this procedure, as we are aware that toothpick has a quite pointy end, and that pointy end will be used to scrape the inner of the cheek, and it will be put on the glass slide before it is observed under the microscope. In order to make sure students are performing well in this experiment, it is crucial for you to ensure that students scrape the inside of the cheek gently with the white of the toothpick, so they are not going to stab themselves with the toothpick. So here, I'm going to show you the correct way to scrape using the toothpick that should be done by a student during conducting this study without causing any injury to the cheek.
Toothpick we're using has a small end or sharper end and a flatter, bigger end. You want the big end to go in your mouth and the sharp end in your fingers. So pick up your toothpick by the sharp end. You're going to put it in your mouth and you're going to go up and down 12 times, scraping against your cheek. Okay, once you do that, you're going to swirl it into the dye, one side, spread the dye out, flip it over, do the other side, and get all your cheek cells spread out. And then you're going to throw that away.
Next, we are going to focus on the permission note. Before conducting a lab, it is advisable for us to provide additional rule for specific situation or setting. The rules must be followed at all times. Each student volunteer must provide a permission note signed by a parent to participate in the experiment. Student must have written the class expectation agreement signed by student and parent before being allowed to do lab work. The signature indicates that the student have read the rule, understand them, and agree to follow them at all times while working in the laboratory. For the permission note that was signed by a parent, it indicates that the parent also have read all the rules and hazard, understand them, and agree to allow the children to conduct the experiment. So, if there's anything happen in the experiment, the parent already noted about all the dangers and hazard that exist in the experiment. If you can see here, this is the example of the permission note that must be given to the student parent and need to be signed by them before a volunteer student start an experiment. Lastly, precaution and cleanup procedures similar to those used in blood sampling should be followed. Similar to the other experiment, when we are conducting an experiment, we need to make sure that all the precaution and safety measures are being implemented. Right after an experiment is finished, it is important to clean up all the tools that are already used in order to make sure that the lab is clean and can be used again. Let's take an example for a procedure to clean the glass slide. Glass slide usually used in most biological experiments, including chick cell experiment. After an experiment involving the glass slide, it must be cleaned. Unless if the slide are broken, then it must be disposed in a sharp container. The first step is by using running water, thoroughly rinse the slide. Allow the water to run down from the top of the slide and continue until all the cleaning fluid, including any additional bubble, has been removed. After that, air dry the slide. If the laboratory have hair dryer, it is easier to air dry it. However, if don't, use a paper towel. Blot the slide until it is dry. Microfiber towels may also be used to dry the slide. For each new slide, make sure that the towel you are using is clean. After a given amount of slide, you may need to change to a fresh towel. And lastly, put back the clean slide into the slide case. Each case normally holds 25 slides. Ensure that each slide is placed correctly. If you try to overload the case with more slide than it can handle, the slide may smash together and fracture.
Okay, so next I'm going to proceed with the second subtopic of experiment with bacteria and fungi, which in this subtopic it includes the microbial culture on how to handle microbial spillage, ways to dispose unwanted cultures and contaminated material, and lastly, I'm going to share on the sterilization steps after work. In handling any microorganism, cultures of microorganisms should be treated as potentially hazardous due to its possibility of being contaminated by pathogens or becoming virulent as a result of mutation. This pathogenic microorganism then may gain access into the human body if a body surface has open wounds or cuts which come in direct contact with the culture, or they will also gain access to the human body if aerosol above the culture are inhaled, or contaminated food or drink is ingested. And because of this, all microorganisms and their culture should therefore be handled with caution. Next, hygienic measures should also be emphasized when working with microorganisms. Students or the person doing the experiment involving the microorganism should wear a protective gloves. Wearing a laboratory gown are also recommended to prevent contamination of personal clothing. If the person have any open wounds or cut on the body surface, those should be covered with sterile dressing before starting any microbiological experiments. Mind that we need to also employ aseptic technique when working with the microbial culture. Before and after work, we need to clean the bench surface with disinfectant as well as wash hand thoroughly, and all unwanted culture should be sterilized and disposed after the experiment. We are also warned that to not use mouth pipetting during the microbiological experiment. Instead, pipette fillers should be used to help transfer the liquid cultures. In the topic of microbial culture, I will share on the microbial source, inoculation of the culture, step to tape and label the apparatus containing the culture, incubation of microbial culture, and the precaution measures that need to be taken care of when doing examination of microbial culture. For the first part of microbial source, we should never culture microorganisms from potentially dangerous sources such as sewage, human mucus, pus, or feces. We also need to use only non-pathogenic microorganisms when doing the experiment. The microorganism can be cultured on agar medium or on any substances on which they can grow naturally, such as bread or fruit. Next is the inoculation of the culture, which the person handling the experiments need to always practice aseptic technique in inoculation. Immerse an inoculating tool such as inoculating loop and knife in 70% alcohol before flaming, and make sure it is not in the reverse order to prevent ignition of the fire. Next, when doing the experiment, we need to be cautious in handling the flame loop. Let the inoculating tool cool down first to avoid killing the target microorganism and also to reduce the risk of generating aerosols. After use, we need to sterilize the inoculating tools immediately to avoid spread of the microorganism and the contamination of the workplace. Next is on how to tape and label the culture. By using the permanent felt pen or wax pencil, label the base of the petri dish rather than the lid, and by this way, we can avoid mixing up of the culture in case the base gets separated from the lid. And we also need to hold the whole petri dish set, the base and the lid together. Then, if necessary, seal the dish with adhesive tape to avoid contamination or accidental separation of the base from the lid. The sealing can be made in a manner so it can allow gases exchange to occur. For the incubation of microbial culture, the culture of microorganisms should be incubated in an enclosed environment such as incubator. During the incubation, the covered dishes should be placed upside down. And most of the microorganisms used in school microbiological experiments, it can grow well at the room temperature. Mind that raising the temperature at 37 degrees Celsius favor the growth of microorganisms to be pathogenic to the humans, and thus this way is not encouraged. As for the examination of microbial culture, mind that any transfer of microbial culture by students should not be allowed and not encouraged. The specimens need to be sealed when being examined, either by examining in tape, petri dish or in sealed transparent plastic bags. Opening the petri dish containing cultures or microorganisms for inspection is also not recommended. If the petri dish containing cultures of the microorganism that must be opened for inspection, the teacher or the laboratory technician should kill the microorganisms by exposing the culture to methanol vapor, which is a filter paper soaked in 40% methanol solution, or which is also called formalin, in a petri dish for 24 hours in a ventilated fume cupboard prior to class inspection. Next topic is on how to handle any microbial spillage. Microbial spillage should be dealt with teachers or laboratory technician. When cleaning up the mess, the person handling it needs to wear protective gloves and laboratory gowns. They are also encouraged to wear masks when appropriate, because it is important that they are not inhaled aerosol cloud formed above the spill. The spillage then need to be covered with a towel soaked in a disinfectant such as hypochlorite. Then the towel should be left in place for 15 minutes and then swept into a suitable container. The contaminated area should also be disinfected properly and thoroughly. In case the skin comes into contact with the spillage, the person needs to wash with the liquid soap and water immediately and seek medical help if necessary. The next slide is I'm going to share on how to dispose unwanted cultures and contaminated materials. The unwanted culture should be destroyed by the steam under pressure, which is also called as autoclave, or it can be immersed in the disinfectant for several hours before disposal. All the apparatus that were contaminated with microorganisms or waste materials should also be treated in the same way before disposal. On the side note of autoclave usage, the use of autoclaves in school is governed by the Code of Practice for Owners of Boilers and Pressure Vessels, which issued by the Labour Department. The autoclave has to be registered with the Labour Department and must be examined and inspected regularly by the appointed examiners. The autoclave should also be operated by competent persons unless the autoclave are granted exemption in accordance with Section 9 of the Boilers and Pressure Vessels Ordinance. And lastly, I'm going to share on the sterilization technique after working with the microorganisms. After each microbiological practice, the bench surface should be wiped with disinfectant immediately, such as 70% alcohol or 1 to 10 bleach solutions. We also need to wash hands thoroughly with liquid soap and water after microbiological work. And for drying purpose, paper towels are preferred and used, and once you have done using it, it needs to be disposed in a waste container with a lid to avoid spreading the contaminated paper towels to other areas. And with that, the next topic will be discussed by my other group mates, so I'm going to pass the stage to Nur Mashita.
Hi and Assalamualaikum. My name is Nur Mashitah binti Ahmad Soan as the third presenter. Now I am going to explain on the special concerns in the study of fungi and molds. Molds are a type of fungus capable of producing toxins, volatile organic compounds VOCs, and other metabolites. They grow in colonies and are disseminated as spores by both wine and water. While mold and other fungi play an essential role in nutrient recycling in nature, they can be a real threat when they migrate into school laboratories. Mold growth is usually visible and often produces a musty odor. It can damage laboratory apparatus and it can also have an impact on health. The first one is breathing problems. As mold grows, spores, cells, fragments, and unstable organic compounds can enter the air. They can produce allergens, irritants, and mycotoxins. Some of these can be toxic, especially to individuals who have a sensitivity to them. Besides, dampness encourages materials to break down, increasing the volume of particles or dust in the air. These particles can irritate the lungs, nose, and throat, especially in a person who already has a breathing problem, asthma, or a chronic lung condition. The second one is allergies. A person with a sensitivity or allergy to any mold-related particle may react. Mold allergies can produce similar symptoms to other allergies, such as hay fever or seasonal allergies. In these two, airborne substances can affect the upper respiratory tract. The symptoms may include a blocked or runny nose, an itchy throat, sneezing, and watering eyes. People with a mold allergy as well as asthma have a higher chance of having an asthma attack when there is mold in the environment, and a higher volume of dust can increase the risk of dust mites, which can also trigger an allergic reaction in some people. The third one is aspergillosis. Some type of mold, such as aspergillus, can cause a serious health problem. Most people can breathe in the spores of this fungus without becoming sick, but people who have a weakened immune system or an existing lung disease can have a severe reaction. Allergic bronchopulmonary aspergillosis, ABPA. This affects the lung and can cause breathing problems. The second one, allergic aspergillus sinusitis. This affects the nose and can involve a headache. Aspergilloma. This can cause a cough, which may produce blood as well as breathing problems. Chronic pulmonary aspergillosis. Symptoms may include breathing problems, a cough, and weight loss. Some evidence from the WHO and the Centers for Disease Control and Prevention (CDC) suggests that people have experienced the following symptoms after spending time in an environment where mold is present, including skin and eye irritation, wheezing, fever, fatigue, nausea, headache, insomnia, mucous membrane irritation, and sick building syndrome. Molds can be formed when tiny free-floating reproductive particles called spores come into contact with water and virtually any organic material. Spores that dwell for 24 hours to 48 hours under these simple conditions can become entrenched, then grow quickly into macrostructures that are visible to the naked eye. Visible mold is often accompanied by a foul musty odors. Mold and other fungi are preservative in outdoor environments where they are usually found in much higher airborne concentrations than in an indoor air. They typically enter the laboratory through openings such as windows, doors, cracks, and on the surface of objects brought indoors, such as the student or even the science teacher themselves. Molds require moisture and organic substrate matter to grow and propagate. Some situations, wet such as damp, humid, and wet conditions, create very suitable conditions for undesirable mold growth in this school. Given that these conditions may exist in our school laboratories, proper cleanup and remediation activities need to be done to recognize the actual or potential presence of mold, thus taking precautions as a science teacher. To solve both the school community health and material-related costs associated with mold and other fungi, as a science teacher, I would remind my students to always isolate the clean-up area, thus preventing unprotected individuals from entering the clean-up area. Next, always wear appropriate personal protective equipment (PPE) or wear a mask when potential mold-contaminated material is present. Other than that, be sure to always remind them to avoid touching mold with their bare hands or skin or breathing in mold spores. Then, take care not to cause mold or mold spores to be dispersed into the air where they can be inhaled by building occupants. Be cautious when checking behind wallpaper and other wall coverings to avoid potential release of spores. Make sure to cautiously remove bag, seal, and dispose of any damaged material such as laboratory apparatus, specimen leftovers, or petri dishes. Moreover, avoid painting over mold. If cleaning surfaces, wet wipe the mold with soap and water or bleach solution and use a high efficiency particulate air (HEPA) vacuum. Don't dry escape mold contamination. Last but not least, avoid running the HVAC system if you know or suspect that it is contaminated with mold. Do not use an air mover where dry materials can be disseminated. So, what are the best ways to address the inevitability of being exposed to mold? According to the EPA and the CDC, the most straightforward way to reduce the impact of mold in a laboratory is to control localized moisture or dampness, humidity in the air, and the amount of dead or decaying material on surfaces. But this isn't possible if all lab equipment or parts because it's impossible to keep water and organic matter off all the surfaces. Other than that, materials known to readily support fungal growth primarily include porous surfaces such as damp cloth that is used after an experiment has been conducted, upholstery, drywall, paper products, and leather. These soft materials are capable of retaining large amount of water and provide spores with an abundant surface area to establish viable colonies. However, non-porous plastics, woods, and composites, have also shown the ability to sustain fungus. That's all from me. Moving on to the next presenter, which is Hakim.
Hello and hi everyone, my name is Hakim and now I will continue our presentation with this part, which is extraction of chlorophyll and the use of flammable solvents. What is the relationship between the extraction of chlorophyll and the flammable solvents? Do you all want to know the answer? Let's check it out. For your information, in order to get the extraction of chlorophyll or chromatography, we need to separate the pigments of plant clearly. So, we need to use the mobile phases of flammable solvents such as acetone, hexane, or ethanol to identify which pigments that appear in the extraction. Apart from that, we should need to take precautions when doing that experiment to avoid any incidents in the laboratory. First of all, when we enter any laboratory, we need to wear the personal protective equipment (PPE) and clothing such as lab coat, gloves, and eye protection. PPE is important because it prepares you for health and safety risks and gives you extra protection in event of an accident or against the elements. Other than that, we need to conduct the flammable solvents such as acetone or alcohol in fume hood to provide a safe environment. The fume hood protects users by containing and exhausting airborne hazards. It does this by constantly pulling room air into the hood and exhausting it directly outside the building through fans located at the roof level. Fume hood sash also provide shielding in the event of an explosion or fire inside the hood. For better explanation, let's watch this video to see how the fume hood works.
In laboratories, a fume hood is a type of ventilation device to prevent exposure of hazardous or toxic gases to secure the safety of laboratory workers. This fume hood device consists of a state-of-the-art controller, receptacles, and safety valves. A fast actuator is able to control the air velocity in case the front sash is lifted, and its performance is verified for maximum safety. Reliability and energy savings are important, so it is essential to have a well-designed device. For usability reasons, the front contains a large side glass sash on top of the front door. When the exhaust air is connected, the air begins to flow inside the fume hood. It enters through the front sash while open and then flows through a baffle to the duct. The fume hood is provided with ideal design baffles to guarantee an optimal and consistent airflow to exhaust of hazardous fume. The structure of the airfoil stabilizes the inflow and prevents the vortex of air. It is designed to create a safety zone in between the front door by separating the space. To allow a correct inflow, it is important to not place any equipment directly on the airfoil. Instead, store them in the back of the fume hood. In general, the recommended height of the vertical front sash opening is 500 mm. This opening height and the side glass sash stabilizes the exhaust airflow and provides researchers with an optimal workspace. For laboratories, the fume hood is an essential equipment to enable a secure and pleasant working environment, and it provides enough space for any type of laboratory instruments. A sensor on top of the fume hood detects movement in the area in front, and it automatically closes the sash if there is no laboratory worker. The automatic front door enables energy savings of 50% and additionally limits the risk of any leakage.
Next, flames should be kept away from solvents or their vapors. We should put the flammable solvents far away from the hot plate and Bunsen burner to prevent the fire. Last but not least, if a solvent ignites in the beaker, cover the beaker with a glass plate to extinguish the fire. If burning solvent is spilled out on the table, use either the carbon dioxide or dry powder fire extinguisher. For your information, there are 6 types of fire extinguisher, which are water, aqueous film forming foam AFFFF, carbon dioxide, ABC powder, deionized water mist, and wet chemical. Each of the different types of fire extinguisher is suitable for different fire classes. First, water fire extinguishers have a red label and a Class A rating. They are suitable for fighting fires involving solid combustibles such as wood, paper, and textiles. Second, AFFF foam fire extinguishers have a cream label and are highly effective on Class A and Class B fires. The foam agent helps to prevent reignition. Third, carbon dioxide fire extinguishers have a black label. They were originally designed for use on flammable liquid fires and therefore have a Class B fire rating. Fourth, ABC powder fire extinguishers have a blue label. They are versatile and can be used on Class A, Class B, Class C, and electrical fires. Other than that, deionized water mist fire extinguishers have a white label and are highly effective on Class A, B, C, and burning electrical equipment. And lastly, wet chemical fire extinguishers have a yellow label and are designed specifically for use on fires involving combustible cooking media such as burning oil and fat. That's all for my part. We will continue the presentation with our next presenter.